And then one time it happens, we're all like, why did you do that? Like, why? And then, yeah.
Listen, it just adds to the comedy of the show, okay?
But it's slowing things down.
Guys, we're a comedy podcast.
We need to be efficient. Okay.
You've now put pressure on us to say something funny in the first 5 minutes.
We always say laugh because we're hilarious.
We are hilarious.
Yeah.
Hello everyone, welcome to Layer Tats, the No Bullshit Science podcast.
With extra bullshit.
I'm Curie.
And I'm Alice.
And she's Demelza.
How are you, Curie?
I'm okay.
I wanted to ask because you normally ask me first, so I was like, this week I'm going to ask first, and I jumped in there.
Very good.
It didn't go anywhere any better than it normally goes.
No.
We're still super awkward.
I just don't have anything to say. I really don't have much to update at the moment.
I feel like I have loads.
Go on then.
Okay, so... Well, should I start at the furthest away thing or the closest thing?
Start at the most exciting thing. No, start with the least exciting thing so then we can end on a high.
Okay, so on my way here, I was walking from the train, and I was walking along and I saw it fly really quickly out of my peripheral vision, and it was coming towards me, and the next thing it was in my eye.
Gross.
In my eye, and I felt really sorry for the fly because it was a really small fly and it got stuck in my eye and I could feel it in my eyelashes, and I was trying to get it out, and I had to stop because I kept walking into things, and then the next thing I looked down and I was playing with my eyelashes and I saw the fly falling out of my eye.
I mean, if it makes you feel better, it probably would have died today anyway.
It died in my eye.
No, but it was gonna die anyway. It's okay.
Why was it gonna die?
Because most of those little tiny flies only live one day.
Okay, but anyway, it went. Anyway, I thought you might laugh a bit more than you actually did, so I'll move on swiftly now. So, my next thing is, my half marathon.
Can I just say that I've been hearing about Alice's foot for about 3 weeks, and I really don't care anymore.
Well, actually,
Although
I was
that.
going to talk about the half marathon more than the foot, but you've just brought it back around to the foot, so you're forcing me to talk about my foot.
I say that, but I do ask you how your foot is.
You do, yeah, because I ask you how your ankle is.
Maybe I do care about your foot.
So for context, I'm running a half marathon on Sunday.
Woo!
Actually, when this is out, I'm going to have run it already.
Woo-woo!
Yeah, so it's my first one, and I'm a bit nervous because I was doing really good. It was my New Year's resolution to become a runner. And it's the only resolution I think I've ever kind of kept to. Had a training plan, was doing great. Went to watch the London Marathon. It was super motivating, even though I'm doing a half marathon. Emphasis on the half.
Hey, it's still a half. That's still loads.
Yeah. Anyway, so then I went for one of my long runs after the half marathon. Felt great. Then I was in the gym the next week, and I hurt my foot, and I haven't been able to run properly since. And that was 3 weeks ago. So I'm a bit nervous.
You'll be great.
I'm really sad because I was on track to do it in a decent enough time.
You're still on track. And also, whatever time you get, it'll be your PB.
Exactly, because it's the only one I've ever done.
Exactly.
And my foot feels fine right now, in case you're wondering.
I wasn't.
Well, so maybe somebody listening was.
Anyway. Do you have any more updates?
After my half marathon, I'm going on holidays.
Again.
I'm going to Greece.
Very nice.
This is for my mom's 60th, although she'd kill me if I just told everybody she was 60.
Although, to be fair, your mum looks nothing like 60.
No, that's what I keep saying. I'm stop being upset that you're 60. You look great.
A life starts at 60 now.
Exactly. So, yeah. So, we're going on a little holiday, me, my mum, and my sister.
Very nice.
Yeah, it's going to be fun. So, you do have something to talk about slightly.
What, that I'm a bad plant parent?
No, I thought you said it was growing new leaves.
She has gotten loads of new leaves. So my plant Gabby, which I don't think we've actually talked about, she's from Patch.
Yeah.
Who...
I'll put her on the Insta.
Yeah, so she's from the same group of friends as Alice's Gloria and Kev. And
Kev's actually from Columbia Road Flower Market, just to let you know.
Oh, sorry.
So just Gloria.
Fuck Kev.
Yeah. No, Kev's a great guy.
How's his new leaf?
It's great. He's a beautiful new leaf.
Yeah. Anyway, we're talking about Gabby.
Yeah. Sorry.
So Gabby's got loads of new leaves, which I'm really proud of because I'd never grown new leaves on any more plants before. Actually, that's a lie. I have an aloe plant who's got some new leaves.
Oh, that's good.
It's also not growing taller, so I think it's dying. But Gabby's also sick.
Oh, why?
She's got brown spots on her leaves, and so I've emailed Patch, sponsor us, so that patchplants.com, Patch on Instagram, could they basically diagnose her and tell me how to save her.
And they haven't replied yet.
Well, they have, and they said it could be overwatering, it could be underwatering, it could be too dry air, it could be humid air, it could be too much light, too little light, it could be the aircon, it could be the heating. So I have to answer loads of questions, and then they'll diagnose her for me.
To be fair, your office is a very climate-controlled environment.
Yeah, but I think she's meant to be okay in those, but let's see.
They're not very humid, though.
No.
So maybe I need
to
humidity.
start spraying her.
Yeah.
But I said to keep their leaves fairly dry.
Okay, interesting.
I did clean her leaves today, though.
Yeah, you need to do that, or else they can't do photosynthesis.
It was like a really good 15 minutes of not doing any real work.
Yeah, nice. But feeling like you were contributing to a living thing.
Yeah.
Yeah.
Okay.
Yeah. Oh, on that note, so you know how everyone, like, well, I don't know, me and you, but I also know a lot of people who are really obsessed with plants. Mm-hmm. So apparently I read this article, and it's because millennials really like plants, and the reason for that is because if we were a couple of generations ago, we would be having children by now. But because we're millennials and we don't make decisions and we think we can do everything, none of us are having children. So instead of having children, we have plants.
Interesting.
Yeah. Isn't that really interesting?
Yeah.
Because it is such a thing. Everyone's so into plants.
But if plants are
like Patch is like, I'd say, thriving.
But that's because they have great marketing.
And because of all us millennials.
But if plants are my surrogate child, I'm fucked.
Yeah, you are.
My future child is fucked.
Well, we'll see.
Anyway, shall we introduce our guest?
Yeah.
Because I know he's dying to contribute to the Patch talk.
About Patch.
Yeah.
Shh, I haven't introduced you yet.
Okay.
No, he's excited about Patch.
Right, so this week we have Ben.
Hi, Ben.
Hello. Thanks
Hello.
so much for having me.
You're welcome.
Thanks for coming.
Of course.
Ben and I, and Silent Demelza, we have an interesting meat story.
Ooh.
A meet cute.
Meet
I'm intrigued.
So I think you'd be better explaining it because it's like your little baby project.
Wait,
Yeah.
before we move
Are we gonna
along,
come back
though,
onto the Patch?
before we move along, do you want to talk about Patch first?
A little bit, yeah. I do a little bit
Go
want
on,
to talk
tell
about
us
Patch.
Okay, who do you have?
Oh God. I've just got so like it's become a problem actually how many I have. Like it started off like a normal
I think
amount.
that's another millennial trait. I think
Yeah.
it's just wanting too much. Yeah.
Just wanting so much.
Yeah.
And like I've got another housemate who loves plants, and she actually brought a lot of plants with her. And then we've got another housemate called Tom who very much tolerates the plants.
Okay.
And he's kind of his rule is as long as I can like, walk without
Tripping
walking
over a
into
plant,
one,
yeah.
that's fine. But I think even now he's kind of like at the end of the limit of what he can deal with these
Right.
plants. He quite likes flowers though, so every now and then he'll bring back a daffodil or a tulip
that's
or something
nice.
like that.
Lovely.
You didn't get like what Kevin was actually. Was it
So
Kevin? Sorry.
Kevin, he's from Columbia Road Flower Market.
Right.
He's a cheese plant.
Oh, sure.
Gloria is from Patch, but she's actually been renamed, so she's a peace plant. So her name from Patch is Pippa. But I know a lot of human Pippas, so I
okay.
didn't want to get confused when I was talking about Pippa, and people would think that I was talking about a person rather than a plant. So I renamed her Gloria, but she's a peace lily.
Oh, okay.
Yeah.
I love those.
Yeah. Oh,
Have you ever had an asparagus fern? I've got
No.
like okay. So if Patch are listening now, no one has been able to teach me like. All of my asparagus ferns have thus far been like born to die, and like I've done everything. They are clinging on to life, but they're kind of like in the ICT unit, or
Yeah.
is that
ICU?
ICU, yeah,
ICU.
sorry, ICT. The ICU unit, that is the bathroom, and I mean they suffer from underwater and overwatering.
These look
humidity.
like they'd be hard to grow though.
Yeah, but I just love them because they look so
they are
prehistoric.
cool.
Like I can.
yeah. You can imagine dinosaurs roaming around,
Apparently they are prehistoric. Well,
leafing through them.
they're obviously a very successful plant.
Yeah.
Because they've survived since, well, like probably pre-dinosaur and whatever killed the dinosaurs they've lived past.
But yet they can't
And my
survive your
bathroom,
flat.
yeah.
No,
Yeah.
no.
And I've done everything.
So sad.
Yeah.
Mate, well, Patch, if you're listening,
Yeah.
Ben would really like some advice.
On
And
the
we'd
asparagus,
like
yeah.
you to sponsor us.
Yes, please.
Yes, please. So who have you got? Who's in your Patch family?
So I actually, like, even though I look at Patch on Instagram every single day, I've actually never had a Patch encounter. But I've got 3 cheeses, like one that we inherited that's
Okay.
been growing for like about 20 years or something from my housemate's friends. And this cheese is it's like
Oh my God,
bigger than this
wow.
table. Like it's like the air roots are probably about like 3 meters long, and they're like
Oh my
curling around it
God.
like this. And and then kind of two adolescent cheese plants either side, and they kind of
I would say mine might be an adolescent.
Right, okay.
Early adolescent.
How big?
I'd
If you
like not we're not talking arm span. We're talking like, well, maybe an arm.
Yeah.
Yeah, maybe like.
Okay.
I think he might be still a baby then.
What do you think?
I think kind of like 12.
Yeah.
Okay, yeah, early adolescent.
Yeah. Pre-pubescent, but not.
Yeah. He's getting there. He grew a new leaf.
Okay, that's good.
Yeah.
Does it have the
And it came like curled outwards.
Oh yeah,
it was so exciting.
Yeah.
It's just finished curling out. But I spoke about this on the last episode because I thought when I saw the new leaf it was all curled in, so it was just literally like a spike kind of. But I got really freaked out because I thought either that's what happens before the leaf falls off and it dies,
right?
Or there was some sort of weird creature living inside the leaf. And this plant is in my bedroom, so I was like, oh God, no. And then I googled it and it was a new leaf. I was so excited.
Yeah. Sometimes I watch like the leaf uncurling on YouTube in like a sped up video.
Oh my God, I'm gonna do that.
Yeah. Or you can like the videos of the calathea, the prayer plants, do it like going up, like if you speed up the video, they're really impressive.
Oh, wow.
Yeah.
Nature's just great. I love
Isn't
nature.
Nature's alright.
Oh my God, what is wrong with you?
Sometimes I just don't like being in nature for too long, you know?
Says the girl who just invited me to go camping.
That's true.
We're going camping.
Camping is a weekend, and there's still toilets there, so we're all
Okay, fair.
My dream is, again, if Patch is listening, this could be made to happen.
Please listen, Patch.
My dream is talking about nature and kind of like how we could actually recreate nature, I guess, in the house, is to have... I worry that the plants, there's a lot of plants, but they're not in the same soil, so they're not communicating in terms of their roots, and I actually would like to have them as more of a network, but then I don't know how to have that without having like a weird trough in my living room.
You would have to have a trough around the perimeter of your house.
The only way is a trough.
It's the only way, I think.
Unless Patch have some other ideas.
Patch, do you have a solution?
Yeah.
Make
My
this
trough
man's dreams come
please.
true. Okay, so have you gotten all the Patch out of your system right now?
Yeah, for now, we can come back to it.
We can circle back.
Yeah,
we
circle
can circle
back.
back.
Okay.
Yeah. Okay, so should we go back to how you three met? Because I'm really intrigued.
So I am doing a PhD in the French department at King's College London, which is this university that we're in at the moment. And I work on contemporary French thought and culture. So contemporary French philosophy and literature and film.
So you're a real PhD.
How do you mean?
That you're a Doctor of Philosophy.
Oh.
That's like the OG PhD.
Oh yeah, I've never really thought. Yeah. I've never really known what that means, like Doctor of Philosophy.
It's you.
I guess. But so
it's like that's all the Greek guys. Like,
yeah. Well, I guess
Aristotle and all those people.
I guess that's where it starts.
I
think
so. Yeah.
But like, I've never really known why it
they
starts.
They are Greek,
there
aren't they?
Yeah.
But yeah, so I kind of only do like contemporary stuff. So like people that are doing philosophy today, basically. And I work in particular on the philosophy of a philosopher called Catherine Malabou. And her major concept her central concept in her philosophy is plasticity.
Okay.
And she's a very interdisciplinary thinker. So she kind of thinks across philosophy, neuroscience, art, psychoanalysis, like all of these different things. And she kind of argues that plasticity has become a central concern of all of these domains. So like in neuroscience, obviously neuroplasticity has become a massive thing. I mean, we're sat here doing this podcast in a neuroplasticity lab, and then in the arts you have the plastic arts, for example. It just names kind of in terms of plastic arts, that's kind of everything that can be like shaped or molded or
literally
or like,
physical things.
Like
clay.
Sculpture, like all sculpture is like a type of plastic art or something like this. And then so I guess for Malabou, she's kind of noticing that plasticity has become this central concern across all of these disciplines, and her philosophy is about asking why that is and what we should do with plasticity, and also what these different types of plasticity emerging across all of these disciplines have to say to each other. Yeah.
That's really interesting. So in terms of, I suppose, so she's looking at the different like definitions of plasticity in different areas, kind of.
Yeah. Oh, actually, I forgot to answer the question, which is how we
Yeah,
met.
yeah.
Sorry, so through me doing this PhD, I'd always thought from the start of the PhD that I wanted to, because Catherine Malibu was talking about neuroscience so much, that I really wanted to actually talk to scientists and get to see what plasticity looks like in the lab. And so I had the chance to do this kind of cross-faculty project, which we called Narrating Plasticity. And this was Sandrine Touré's lab was the lab that very generously decided to collaborate with us on the project. And so the project brought together an artist called Amanda Deutsch, whose birthday it was yesterday. So happy birthday, Amanda!
Happy birthday, Amanda!
So she was kind of the plastic arts component of this. And then I guess I was coming at plasticity from a philosophy angle through Catherine Malibu, and then plasticity in the neuroscience lab. And so it was a very open project and just about kind of creating dialogues across these disciplines. And so the lab came to the artist studio and Amanda came to the lab. And yeah, it was just about kind of, we wanted to know that if there is a certain type of or definition of plasticity in the plastic arts, this kind of idea of moulding. And if the word plasticity comes from a kind of aesthetic definition of meaning to shape or to mould or whatever, and if this word is used in labs as well, like how much overlap is there with that? And are these similar types of plasticity or two different types of plasticity? And how do they challenge each other? So yeah, that's what it was about. And it was a kind of 6-month project. And we really wanted to kind of go further with it as well after that, and hopefully in the future, like, we will find new avenues to explore with it and opportunities to explore it further. I know Amanda is still creating a lot of kind of brain neuroplasticity inspired artwork, and she's going to be presenting it this summer as well. So in terms of Amanda, this has been like a big trajectory in her art. Yeah,
that's so cool. Is she based in London?
Yeah, she lives in North London.
Okay.
Yeah. Oh, and that's another thing to mention. Like at the start of my PhD, one of the things that led to this project was that I also had a personal experience with my brain. I'd started doing my PhD on plasticity and the brain in philosophy, etc. And then 2 months into it, I found out that I had a benign brain tumor and needed surgery. And it was really weird. I was on a date with a guy called Sean, and I've told this story like a million times, but it's like impossible not to tell it if we're going to talk about it.
Of course.
I was on a date with a guy called Sean, and we were kissing outside the pub. I'm going to say it very, very quickly.
Go for it.
Because it's really boring for me now. But
boring for you, but like I'm really interested. Continue.
So in the pub next to where the Lion King is, it's called the Wellington.
Oh, yeah.
And we were like snogging at the end of the day, and then these homophobes came past and were like shouting at us, and like we'd had a couple of beers, so I shouted back, and then they beat me up basically. And then I went to hospital, and they CT scanned me, and they saw that I had hydrocephalus. And then they did a few more scans and realised that was because I had a benign brain tumour.
So it was nothing to do with getting beaten up?
Nothing at all.
But it was like a byproduct of finding... Oh my God.
Yeah. It was just like completely incidental fine. So I went on a date and came home 2 weeks later having had brain surgery. Oh, it's crazy.
It was weirdly, in a way, a blessing in disguise.
It was a blessing in disguise.
That's mad.
And so the operation...
And had you gotten any symptoms or anything? Had you been having headaches or anything?
You know what? Like the odd thing is that literally I've never had... One of the things about... I've just never had a headache in my life.
Oh my God.
I'd be like, if everyone was like, oh my God, I've got such a bad headache. And I'd always be like...
Not even when you're hungover.
To be honest, no. Okay, my hangovers are nausea hangovers.
Okay,
I never get
that's bad.
I kind of prefer a headache hangover.
Yeah, I would too.
Yeah.
I've had both, and I definitely prefer the headache.
Yeah.
I hate being nauseous.
Oh, me too. It's the worst thing in the world. Yeah. Absolutely hate it.
Oh, it's awful.
But yeah, like before, when people would be like, "Oh no, I've got such a bad headache," I'd always be like, "Oh, me too." But I kind of didn't.
Did you ever want someone to give you a headache just so you knew what it felt like?
Well,
this is like Curry's pursuit for a bruise.
Yeah, I don't bruise.
Curry really wants a bruise. She doesn't.
Oh, you've never had a bruise?
No, I do, but it takes a lot to make me bruise.
Oh, right.
So I'm always kind of jealous when people have these really obvious bruises.
Yeah.
And my sister has never had a nosebleed. So
really?
She's always wanted to know what it feels like to have a nosebleed.
Oh, that's
We'll
not
often punch her in the face, but she won't. She doesn't go for it.
Well, you're being a good sister.
Yeah. But like, I've never fainted. and I really want to know what it's like to faint.
I've never completely fainted, but I've gone.
Yeah. That
I've almost like I've gotten that kind of rush, but I've sat down or like lain down, and then I haven't fainted. But I felt like my head was emptying.
Yeah. I've come close to fainting. Should we talk about your history with science then?
All right.
Yeah.
Briefly, yes.
Yeah.
Like a brief history of my relationship with science.
Okay, so why did you draw my attention to it?
Because you hadn't noticed Imelda saying 1 minute, so I was trying to let you know discreetly.
Let's talk about your relationship with science then.
Okay.
But it's interesting because we kind of brought you on as our lay guest, and it's kind of like, oh, but Ben is super lay because it's humanities, but actually your work has overlapped so much with science that you're actually going to be quite knowledgeable.
Oh, I don't know.
Maybe.
We'll see.
But so
maybe not necessarily, but like I feel like you're super interested.
Yeah. Oh, I'm very interested. I'm very open.
Yeah.
So what was your relationship with science like at school? Or
did you do any subject in science? Did you?
So I did like GCSE in I did like biology, physics, chemistry. Like one of my major things is that I wish I could go back and do them again, because I think at the time, like, everyone's very like hormonal and like this kind of stuff, and like there's everything else is interesting, and then I feel like I feel like those sciences are so different though, like. And I'd have liked to have known how they overlapped a little bit more, I think. Because I was learning about electrons in chemistry, and I couldn't quite figure out how that related to like cells in biology, or how that related to like the solar system. I mean, these are very different scales, aren't they?
Because this is really interesting. Because before we started recording, when you were asking us about like DNA and chromosomes and things, that's like that. Like you really wanted to know how they related to each other. And now you're like, I want to know how everything relates to each other.
Because I guess like all of these things were talking about particles. So like chemistry was talking about its own type of particles, and then in biology I was like, everything's like a blob, isn't it? Like, and then biology was like drawing blobs on blobs. So I was kind of like, is that blob?
Is this the same blob that I drew in chemistry?
Does that blob translate?
Are these blobs related?
Yeah, I just wanted someone to say, okay, that blob's made of these
like,
blobs.
Tie them together.
Yeah.
But how interesting that that's what you wanted to know at school, and now your PhD is connecting relations and like connecting one blob to another blob.
To be honest, when I was in primary school, I think because we weren't doing any like, I guess we were doing like vague science, like whatever science you do for stats. I can't really remember what you do. I guess like learning what, I don't know, temperature, like and that kind of thing. But and I think like before I went to secondary school, all I wanted to do was like get in a lab and like and like, because I'd seen on like Grange Hill like the Bunsen burners and like that kind of thing, and I was like, oh, that's gonna be so cool. And then I went to secondary school, and actually I really loved engineering as well before I went to secondary school. And then I went to secondary school, and it was like the total opposite actually. I suddenly what I was really drawn to were like languages.
Okay.
Instantly, like French and I did French and German at school, and I loved those. Loved it, and I'd never really done anything like that at primary school, and so it was a surprise for me actually that I didn't enjoy science as much in the same way that I thought I would. And then now that like I guess I've had the chance to put those interests together, I think, and return to science a little
and maybe
bit.
It was like timing as well. Like maybe I don't know. You know the way you're saying now, I'm like, I wish I could go back and do it, kind of thing.
Yeah.
But maybe just. Or the way it was taught could have been anything. Right?
Well, that's what I was thinking, because I wonder if, because the thing that you were drawn to was them being in the lab and using Bunsen burners and stuff like that, right? So you go to secondary school and the languages are taught in a very practical way. Yeah, you do your speaking and all this kind of stuff, and you generally learn through little activities. You get into science and they put a textbook in front of you, and you just learn out of a textbook.
Yeah.
So now you've completely lost that practical element of science, apart from the odd experiment maybe once a week.
Right.
So
and at the beginning, the experiment is heating up water.
Yeah. So I wonder if that's what put you off it, because this practical element of it's gone.
I honestly, and I think it was, I think that's very true. And I think also I always used to like hear like my friends who were like bilingual or something like that, and I think I just always it was as simple as I thought it was really cool when I heard them speak, and I thought, it's so cool that they can change, they can switch, that they can exist in like two different ways of like speaking and two different ways of thinking, or more than two ways. And I think that's why you
changed.
And
can you speak fluent French?
Yeah, I can. So I did my undergrad. I did French and German as well. Kind of continued that from school, and so I get to use French quite a lot in my PhD, but not so much my German, which is a shame.
Yeah.
But yeah,
that's really cool. You're doing your PhD in French philosophy.
Yeah.
Modern French philosophy.
Yeah.
Do you have to actually speak French for a lot of that? Like, is a lot of it in French? Are you reading it in French?
The texts that I'm reading are in French.
Okay.
But so I write the PhD in English, but the texts are in French. And then, yeah.
Interesting. Cool. That was just a burning question that I was like, so is he going to write the whole thing in French or what's going to happen here? So yeah, that's all clear in my mind now. We can move along.
Okay, cool. All right, well, so should we talk about some science then?
Here we go.
Yeah.
Let's do it.
Yeah. Do you know what? So I went on a mission to find something for today because I really wanted to find something. Well, first of all, it's really interesting because remember ages ago you asked me about the 5-2 diet.
Yeah.
And you were kind of like, oh, are there any books you could recommend? And I was like, well, not really books because we don't use books in science. We use papers. And you were like, oh, I forgot about that. And I thought that was a really funny, interesting comparison between the two. Anyway, so then I was looking for these papers and I asked you a few days ago what I should look for. Or is there anything that you'd be really interested in talking about? And your reply to me was...
I enjoyed this.
Gay dating, Celine Dion, drag, or RuPaul.
Yeah.
And so I really tried my hardest.
I wish someone has written scientific papers on RuPaul.
You can see what state I was in that night. When I
Fantastic.
I did search RuPaul and drag in Google Scholar and nothing came up.
Oh,
I also searched Celine
really?
Dion.
I'm surprised at that, actually.
Yep, nothing came up. I searched Celine Dion in PubMed and one paper came up.
Oh, right.
But it was just where they showed someone a picture of Celine Dion.
Oh, and what was the reaction?
I don't know.
Joy.
Ecstasy.
Utter joy.
So then I tried to check it somewhere else. So then I started Googling. I'm trying to find the papers I found. I did some Googling and I was searching homosexuality brain, homosexuality neuroscience, same-sex dating. And I was trying to find something and I just really couldn't find much. And I thought there was a couple of links that I sent to these guys thinking, what do you think about this? One of them was top, bottom and versatile anal sex roles in same-sex male relationships, implications for relationship and sexual satisfaction. And I was like, guys, this is a bit too much. And we were like, maybe, let's see. So then I found another one that was a little bit more tame. And this one was a picture is worth 1,000 messages, investigating face and body photos on mobile dating apps for men who have sex with men.
Great.
So I didn't pick this one.
Oh, okay.
Curry, you should have done that one.
So the reason I didn't pick it is because you're a humanities guy.
Yeah.
And
right, okay, it wasn't...
we wanted to give you something that you're not used to.
I wanted to challenge you.
Yeah, okay.
So I wanted to pick something that was hardcore.
Okay,
science.
I'm glad, yeah.
And by the way, this has completely messed up my recommendations on Nature. So Nature being the best science journal out there. And when you're on the Nature website and you're looking at articles, it pops up at the bottom recommended, read this article.
Okay.
And normally it's articles...
Based on what your searches are.
And normally
so you can imagine what Curie was just searching.
Oh, okay.
So now all the articles she's got coming up are
normally it's very much related to neurogenesis and things that are really relevant to my work. Now I'm getting lots of recommendations on anal sex.
Okay.
It's okay.
Well, enjoy.
So the paper that I've picked for you today is quite a big one. It has been in the news. It's been in the news for quite a while because I don't know if you remember, I brought this up. Oh, you're right.
Oh, yeah, sorry. Just...
I brought this up last year and the paper hadn't been published yet. And then it came up again in the news recently. And I don't know if you've seen it, but it's about pig brains being brought back to life.
Oh, wow.
Yes. So that's the
headline.
headline.
Oh my god, okay, I'm excited about this. Shall I get a pen and paper?
Oh, I love it.
Or
okay.
You don't have to.
I'm going to.
Okay.
Just in case.
Okay, do, yeah. Because sometimes a diagram is helpful.
Yeah.
That's true.
I agree.
Even though everyone gave out to me last week when I was drawing my diagram, but, it got 24 likes on the Instagram. That's up there with our most likes we've ever gotten.
That's not a lot of likes, though. We need to do better.
Perfect.
We're ready.
So
this is a lovely notepad, by the way.
Oh, thank you so much.
It's really nice. Yeah, I like it.
It's just miscellaneous.
It's good. It's great. Just a few thoughts.
Just a few little thoughts.
So the headline in all the newspapers are that pig brains have been brought back to life. This is of course a gross exaggeration. We have not got zombie pigs roaming the streets anymore. Oh,
Anymore.
Anymore. It's okay, guys. They're all gone. They're all gone.
So this paper is called Restoration of.
Yeah.
This paper is called Restoration of Brain Circulation and Cellular Functions Hours Post-Mortem.
Okay.
And the PI for this group is called Nenad Sestan. And when I first saw it, I thought his name was Satan. And I thought that was really fitting.
Oh, God.
What's his name actually?
Sestan.
Sestan.
Yeah. Okay, so I'm going to talk you through this paper. Feel free to interrupt me at any time.
Okay.
Yeah. Ask all the questions that enter your mind.
Yeah. So this paper does get really sort of into it. And so I'm going to skip over a lot of details. So if the author of this paper happens to be listening, don't crucify me.
Can I just say the first note that our guest is after writing is literally zombie pigs.
Which don't roam the streets anymore.
Yeah.
We just
No longer a threat.
Yeah. They're all gone. So the main concept behind this study was that mammalian brains, they tend to be quite big, and they also need a lot of energy. But because of this, it also means that we're really susceptible to damage. So things like anoxia, which is the lack of oxygen. So what would happen if you had a stroke, for example? And stopping of blood flow, we're really affected by those things because our brains need so much energy and they're big and blah blah blah.
And they're very complex.
Very complex. So lots of these studies in humans and animals, what they have shown is that the electrical activity in our brain, our oxygen sort of stores in our brain, and our consciousness are lost within seconds of us losing oxygen supply. From interrupted blood flow. And then if you go on a little bit further, our energy stores from our brains are lost within minutes. So they're kind of saying this damage to our brain is almost completely instant. And that's how important it is that we keep our brains well oxygenated and the blood flow going. And they think that at the moment these are pretty much irreversible. Once you get these changes, that's it. Your brain is dead. There's nothing we can do.
And that's why, isn't it that way, like when you've strokes and things like that, time is like everything.
Everything, yeah. But there are also a few handful of experiments that are kind of showing that maybe this isn't the case. Maybe just because we've stopped the oxygen flow and your brain has stopped functioning, maybe that isn't quite the end. And some of these have been that we seem to be able to do some experiments with slices from brains of dead animals. And we can even seem to take electrophysiological recordings. So we have our brain functions through electrical impulses, electrical currents, and we still seem to be able to take recordings from those even after the animal has died. Another one is our mitochondria. So they're the parts of the cell that provide energy. So the classic line from any textbook is that the mitochondria is the powerhouse of the cell. The mitochondria.
He's writing that down.
Powerhouse.
Yeah. So the powerhouse of the cell, the mitochondria, seems to...
It's like a battery.
Okay.
Isn't it?
Yeah.
Yeah.
It provides the energy. It seems to remain functional for up to 10 hours post-mortem in human brains. That was another thing. Also in cats and macaques, even 1 hour after complete stopping of blood flow to the brain, they seem to be able to recover functionally afterwards. With humans, if you've had a stroke, a thrombectomy, which is removing of the blood clot, can actually result in sort of reversing a lot of the negative effects of a stroke up to 16 hours after it's happened. So these are all kind of things saying that, well, actually, maybe once we have lost oxygen supply and blood flow, that isn't actually the end. And there is something that we could maybe do to reverse this or bring it back. So that's what these guys wanted to do. So, to test
Any questions so far?
No, I think we've got the powerhouse of the cell.
It's so funny. That is literally the thing everyone remembers. Like, you will not forget that as long as you live.
Can you draw a mitochondria?
Yes.
What it
Oh,
looks
I'd
like?
love to draw a mitochondria. Okay.
Okay, so for the people that are listening and cannot see what Alice is doing.
Sorry, it didn't need to be this big.
It's an oval, and then on the inside it's like a squiggly line.
Don't worry, this will be on the Instagram.
It will be on the Instagram. It almost kind of looks like the inside of a pomelo. Or
I don't know what a pomelo is.
Some sort of exotic fruit.
Yeah.
Okay, yeah. So, and then this is empty space.
Maybe like a squished pomegranate.
Empty, yeah. Or like probably like some fluidy stuff, but it's pretty much empty.
And this is in the cell.
Yes.
That's
so this is if you can imagine, this is like an egg, and I've sliced through it.
Okay.
Yeah. So this is the inside of it.
Yeah. So that's the powerhouse of the cell.
Yeah.
That provides the energy.
Okay.
So what these guys did is then they developed a...
Can I just do one thing on the diagram?
Yes.
So can I just illustrate? So all along the membrane, there's like these little like machine apparatus-y kind of things, and they're what's making the energy. So like the more surface area you have, the more energy you're getting out of it. Mm-hmm. So then if you've got more of these little spiky bits, then you've got more energy. And exercise increases the spiky bits. Okay, done.
I remember from GCSE biology actually, they said if you don't know the answer, put surface area. Yeah.
That is
and
literally
I've always remembered that.
Surface area is the answer here. Yeah.
That's actually true for a lot of things.
Apparently that's the reason why a walnut looks like a brain, is that they both developed the same way
mechanism of getting bigger surface area
within a shell, basically. Yeah.
Okay, can we not make diagrams a weekly thing?
I like the diagram.
This is an audio platform, Alice.
I know.
Okay, moving on. So what this group did was they basically developed this device and procedure that they can do to reperfuse the brain, basically. So perfusion. Bear with me 1 second. Perfusion is basically the movement of fluid through channels to a tissue or organ. So it's like just replacing the fluids. And they did this in 6 to 8 month old pigs, 4 hours post-mortem. And these brains, they actually got them from food processing facilities, and these brains would have been thrown away. So these guys took it from them and used them for their experiments. It said in the back somewhere that they went through almost 200 brains.
Wow.
Yeah.
Why? Did they have certain criteria for the perfect brain?
No, so it was setting up the device, like testing their surgery procedures and testing the device.
Yeah.
And so all the work they did before this paper and including this paper
yeah.
was about 200 pig brains.
Right. But these pig brains wouldn't have been used for anything else otherwise?
No, they would have been discarded. Okay, so the way they were this paper is interesting in the fact that they do show things, but it's beyond the fact that they're showing their result, which I will go into. It is also kind of advertising this device and technique that they've come up with. So it's a big test of does this work, what can it do, and now on look at this product and device and technique that we've made that others can now use. So they wanted to test how it performed compared to they had a pig's brain that was perfused with a control substance. They had which is basically just fancy water. They had a pig's brain that was perfused with their perfusate, which we're going to call Bex, because their company and their device is called BrainX.
Ah, okay.
So it's going to be called Bex. And they also had a brain that was not perfused and was just kept in room temperature. And they have a brain that is that was flushed out, so just everything was sort of the normal procedure they do after death was done as normal, but this was 1 hour after death. So this was a very fresh brain. And they were going to put all these brains through the same procedures and then compare them. So the first thing they did was they wanted to see if the circulation and sort of the vasculature within the brain was intact and functional. First they needed to test how long they need to perfuse these brains for, and they kept it to 6 hours. This was because the brain that was perfused with the control perfusion, that one started to degrade really badly after 6 hours of being perfused. Whereas their solution, even though it kept the brain intact for that whole 6 hours, that was sort of the limit if they wanted to be able to compare these two brains. Right, so what they found was that so this machine, it basically mimics a heartbeat. So it mimics the heart pumping blood through the brain. And the brain that was pumped with their Bex formula actually had a really good blood flow through the major arteries of the brain. And this was much better than the other types of experiments they were doing and the comparisons they were looking at.
Question.
Yes.
Does the machine pump blood?
Yes.
Okay, so what they've done is they've bathed the brains.
But it's not blood.
Okay.
Yeah.
But for all intents and purposes, it's blood.
Yes.
So they've bathed the brains in different things.
Mm-hmm.
One in Beck's.
Yeah.
One in fancy water.
Yeah.
One in nothing.
Yeah. And one was just fresh.
And one was just fresh.
Yeah.
And now they're putting them through this machine to see if the blood will flow through in the right way.
Yeah. So what? So the control, the fancy water and the Beck's, they're going through this machine. The one that's left as room temperature and the fresh one aren't.
Okay.
They're just extra comparisons to see the difference between one that's been perfused, one that's not being perfused, and then because the fresh one is going to be the closest we have to an actual brain.
Yeah.
They're also comparing that because by the end of the 4 hours post-mortem and the 6 hours perfusion, this brain has now been out of the body for 10 hours.
Yeah.
At this point, it should be very unusable.
Yeah. And what's in the Beck's perfusion again?
Okay, so I took a picture of the things that were in it, and I don't know what any of them are.
I bet you there's glucose. Is there glucose?
There is glucose.
I knew there'd be glucose.
So, okay, I knew you'd ask this.
There's always glucose.
And I was really hoping you wouldn't. So, there's something that helps the gas exchange called Hemopure, which I think is just hemoglobin and something. The metabolic compounds were glucose and sodium.
Makes sense.
Then they had
sugar and salt.
Then they had lots of protective agents.
Lucky brain.
Now, the protective agents. So we start with hexahydro-2-amino 1-HTNol-3,4-D imidazole 4-pentanoic acid 2-aminobiotin.
Got it.
I'm also not going to read the rest of them because they're all like that.
Let me see if I know what any of them are.
So blood flow. So they were looking at the vascular function, and they had very good flow through the major arteries of the brain, so these don't seem to be degrading. Also, they tested capillary flow, so the much smaller vessels, and they just wanted to check that if these were refilling well or not, and they were, which is great. And then they also wanted to see whether the vasculature in the brain was response to a pharmacological intervention. So at that point, you're kind of thinking, that's kind of crazy to give a drug to something that's technically dead and see if it responds. And actually, it did. So they administered this drug, which increases blood flow, but it maintains pressure, like the pressure in the arteries. So the way it does is it just helps gets the vessels to dilate a bit more, and actually that dilation did happen, and the blood flow increased.
Wow.
What was the drug?
It's called nimodipine. Next, they wanted to see how is the actual tissue integrity staying. So, like I said...
What do you mean by tissue integrity?
So the actual, how well is the brain staying in its form and staying as a brain?
Okay. So not melting or turning into jelly.
Exactly.
Yeah.
Or just fading away. And like I said, I think it's clear now that this is kind of them selling their product because they're comparing this Beck's this Brain X technology against these other measures. So they did...
It's like one of those adverts, isn't it, where they've got like the washing up liquids on one side and the...
Like Vanish.
Yeah. And one of the plates comes out and it's still got a little bit of like bolognese sauce on it or something.
And so they first tested the integrity by imaging it with MRI. And under the Beck's condition, basically the structure remained intact. All the brain structure remained intact. They didn't really see many changes, but within the other brains, they didn't see this. The ventricles had started to collapse. It was started to deteriorate, and there was a big, and this is indicative of decreased water content, which they're pumping this with fluid. You'd kind of expect that. So that's good. The other thing they saw was they wanted to look at really susceptible areas. So the most susceptible areas of the brain to damage when you have a stroke or lack of oxygen is the hippocampus, the neocortex, and the cerebellum. And all three of those areas, again, in the Beck's brain, was preserved really well. So that's great for them as well.
Go on, Beck's.
And then next, what they wanted to see is they wanted to check the functions of the actual neurons. Are there still neurons there? Can we still do any work with them? What's going on? Are they dying or not? So they used a staining to now look at this, and they found that in the Beck's brains, the neuronal structure was intact. There wasn't barely any damage. It was exactly how it should be, unlike the other brains that they were looking at.
And how are they looking at the neuronal structures?
So they would use staining. So it's called
staining.
Yeah. And then also, sort of, not only that, but it was also the the number of neurons had also barely changed. So the actual density of neurons that they were seeing in the brain, that was pretty much what it should be, and a much higher than the other brains which have started to degrade and break down and all of that. They also looked at cell death, and these Beck's brains had a much lower amount of cell death compared to the other brains. So far it's looking like this Beck's is keeping these brains nice and alive, which is good.
And remind me, what is the time period of this that they're measuring the cell death or that kind of thing?
So the brains are 4 hours post-mortem, and then they perfuse them for 6 hours, and then they're looking at it. So the brains have now been out of the body for 10 hours.
Okay.
So next they looked at other cell types within the brain. So they looked at two. So they looked at microglia, which are the immune cells of the brain. And they also looked at astrocytes. And astrocytes sort of regulate communication between neurons, and they provide nutrients to the neurons as well. And again, same sort of story. You can see where this is going. It was pretty good in the Beck's brains and not so much in the other brains. And they also sort of looked at, are these cells sort of still functioning in a way that they are kind of giving a response, and when you provide them with some sort of agent that might cause some sort of response by them, and they were. So these cells were still functioning despite this brain no longer being living. And then they also wanted to see, okay, are these individual neurons actually functioning? So then they did a thing called whole cell patch clamping, which is basically a way that they can stick a little probe into a single neuron and measure its electrical activity.
Wow.
And
wasn't Rob talking about patch clamping?
yes,
Is that what he does?
Yeah.
I remember
Yeah.
that. Patch clamping.
It's really boring.
I remember him describing it for way longer than you just described it
Yeah.
for.
And to be honest, I've tried to explain it. I explained it in one other episode before, and apparently Rob was very unhappy with how I described it then.
So-
and he's probably going to be very unhappy with how I explained it just now.
He's going to be
But
extremely
literally,
unhappy. I
it
feel.
is that. You get a cell, you kind of poke it with a probe, and you measure the voltage.
Could this be also some subliminal patch?
Oh, yeah.
Messaging as
Yeah.
well? Yeah.
Yeah. Patch, sponsor us.
Oh yeah, patch.
That'll be the perfect way to come back to it.
It will be.
actually.
Oh,
Yeah,
and circle back around. Patch pants.
Patch clamping. Yeah.
Patch clamp. Patch patch.
And basically, they found completely functionally viable neurons in the hippocampus, which that you wouldn't expect to see electrical activity like this. They do kind of explain it and go into all the numbers. I don't know electrophysiology that well, so I don't understand it, so I'm not going to go into it. They then finally, but when they look at electrical activity on the surface of the brain, using sort of electrodes and sort of ECG type technology to see if there's any sort of global electrical activity within the brain, they couldn't find any. But on a single cell basis, they could.
Right.
So it just means that the neurons are functioning, but there's no network of neurons.
Yeah.
And then finally, they wanted to look at...
But isn't that what you need?
Well, yeah. So their aim of the study wasn't to bring these pigs back to life.
What is the?
To see if you can maybe preserve the function a little bit longer afterwards.
Is it too soon to ask what would the relevance of that be?
Yes, because I'm going to go into it.
Okay.
Yeah.
Okay, sorry, I'm getting ahead of myself.
I have one more result to go through, and then we can discuss.
Okay. Can't wait.
So the final result is that they wanted to see, okay, so we've already seen that there's a few cellular functions. What about overall just metabolism of the reactions that are going on? Sort of indicating that, general cellular functions within the brain might still be happening. And so metabolism is any sort of life-sustaining reaction. So there's three things that metabolism is. One is converting food into energy. One is converting food into the building blocks of your cells, proteins, blah, blah, blah. And one is sort of eliminating waste. That is metabolism. And when they looked at that, they saw actually, yes, there was this resurgence of global metabolism in the brain that was intact in these Beck's perfused brains. compared to the others. So this is great. So what basically, in summary, their main conclusion is that the large mammalian brain sort of retains underappreciated capacity for restoration of microcirculation. So these small scale resurgences in function that we didn't think happened. And they've now provided, with this BrainX technology, they've provided a platform to carry out more functional studies in intact brains, which was something that we didn't really think we could do. Rolling back to your question, Alice, the relevance of this is this now might open up a new way to maybe prolong someone's life or function after an injury, or if they, mostly it's with strokes. So if they had something that caused the oxygen to stop to their brain, like a stroke, and so now do we have a way that we can sort of reverse those effects or prolong someone's life or health after the fact?
Okay, but
Big leap.
They'd be brain dead though.
Well, it wouldn't be using this technology for that, but this now opens up a platform that we have a way to study what's going on and maybe experiment with different ways that we could do this.
Okay.
So yeah, so that was that, and then so this paper is the reason why everyone thinks that there's zombie pigs roaming the streets, and that we have now brought pigs back to life, which, as you can see, we have not. And actually, if you read the interviews by the researchers, they're very clear to say we were not trying to bring these pigs back to life. And I read part of it. I haven't read.
But they wouldn't have resisted it if they had done.
Well, so they said they were making a very conscious effort, despite they knew the odds were low, to make sure that there was no reanimation of these cells of the pig brains. Yeah.
Okay.
So they said
on ethical.
Yeah, exactly. Yeah. So if at any point the temperature of the brains got too high, to a point where they thought that consciousness might come back, they would cool the brains down again.
That's super interesting.
Yeah.
Oh my god.
Yeah. And then there's
because that would have been traumatic for the brain, I guess. Yeah.
And then the closing statement of this article that I was reading about this was kind of saying, does this mean that we now need to reevaluate what is considered brain dead, or what is considered death for animals and humans? And this brings up really huge ethical implications now of what is considered death. Yeah. How can this technology be used? Can we actually start bringing people or animals back to life? Which we are very, very far away from. This paper isn't in no way saying that we've brought something back to life, but we have brought back activity that had been lost within the brain, which is huge. And it's really exciting.
That is really exciting.
Do you know what? Like, I'm not going to lie. I know I'm a scientist, but there's very few papers that actually get me quite excited. But this paper, I'm going to say I was actually really excited while reading it, because I think this is really fucking cool.
Absolutely. Like, and the fact that they were ready for the brain to heat up, as in they had procedures in place, for me suggests that they were kind of half expecting this to happen.
Yeah.
You know?
Yeah.
And that's super interesting to think. Like, I think it's great that that those kind of ethical considerations are obviously taken into place before knowing what that could ever mean, that a brain without a body could like regain consciousness and what sense it would have of itself if it did regain consciousness.
So I would say that was something that was worked into ethics. Like, they wouldn't have gotten away with getting that far without having something in place to stop that from happening.
Yeah.
So,
would you say?
I think I read that they actually didn't need a lot of ethical approval for this because of the way they obtained their brains. They didn't obtain them from living animals.
Yeah, they were killed.
They were already killed. So the ethics, I think, wasn't actually too complicated for this. But they did mention that it does mean that if this device gets rolled out and it's used more widely, there will need to be serious controlling on how this is used. Because if they do use it on, say, brains within intact animals, and they can bring back some sort of consciousness or any sort of function within the brain that might allow the animal to be aware of what's happening, then you're right, it's a really traumatic experience.
Has there been any kind of research on how a conscious and alive brain might operate without, if it was being supported, say in a science fiction context, how it would operate? Because before we started recording this, obviously Alice, you were telling me about your research into nutrition, and we were talking a little bit about Julie Ender's book, and she talks a lot about the gut being like the second brain and this kind of stuff, and like the nervous system being dispersed out through the body, and it not just being a kind of just located in the brain. So like, how much does the brain rely on the body, I guess, to do its thinking, and like what kind of from your perspective, researching brains on a kind of like day-to-day basis, like what do you have any like thoughts on like what kind of what a brain without the body could do? Or think
I think
about?
It's very generous that you say we research brains, because we don't. We research one very, very tiny part of the brain. I don't know, is the honest answer. I think
Did you say what would a brain without a body do?
Yeah, what would it do? Or like think, and like say if it was, cut off from its second brain or its like gut or something, and stop receiving those signals.
Yeah.
Like what kind of quality of consciousness would it have?
Is it so like existential and like, oh my god, what would the brain think about if it didn't have a gut? And
I'm trying to imagine it, and it's like a pure panic. For
I feel panicked now too. I'm like, oh god.
I just think, I don't know. I'm kind of like, if you don't have a gut, I mean, you're not going to lose consciousness. I don't think the gut's going to contribute to consciousness.
But it contributes to being like, you've no energy without your gut.
Yeah.
You've no, like, unless you're being bathed in Bex.
Does that just mean that you don't have the energy to express your consciousness?
Or does consciousness require energy?
Oh god.
Oh god.
I guess like the story that's like in the pop science press at the moment that keeps coming up, that kind of like this made me think of, is that guy who wants to have the head transplant.
Yeah.
I think it's in Russia.
It's China? Is it Russia?
He's Russian.
Okay.
But it's done in China. Okay, am I just assuming that China has really lax ethics in all areas?
Well, they did have CRISPR babies, and
that's true.
Yeah.
What I find interesting about it is that, like every time, like in a lot of these interviews, they've been asking scientists, what about this head transplant? And the scientists have said, well, actually, it's a body transplant because like his head, the head bit is the thing that's going to stay the same, and the rest is going to change. And and all of these scientists have kind of said, we obviously don't know what's going to happen, but he risks, and then they use this phrase, which is a fate worse than death. Like the putting someone's head on a different body and putting it into stimuli that it's not like used to, or those connections not working, but the brain still accidentally being conscious. That in some way this would be worse than death, and that haunts me. That phrase.
That's very haunting.
Yeah.
Yeah, because he would have no muscle memory of anything. He would not know his body at all.
Yeah.
But he'd still be conscious.
Yes.
Okay, so
back to zombie pigs.
Yeah.
Right, back to the pigs.
Any other burning questions or closing statements or other things for us to think about and get panicked?
So when you were saying talking about the patch clamping, you were saying why? So like the individual neurons were able to be stimulated, but networks no.
Yeah.
And so why is it that they can't stimulate the networks yet? And do you think there's like hope for that, or is that just something that is infinitely difficult?
Did they give any argument, or not argument, but did they say why they thought that they couldn't, even though they could like get a reaction out of a single neuron, why they couldn't communicate?
No, so actually they do specifically state it's unclear why they couldn't get this global activity.
Interesting.
But it's their device is in really, really early stages. They're going to need to do a lot more experiments. And to be honest, I guess you can have this activity in one neuron, but then to ask that neuron to then form connections to millions of other neurons and those neurons to form connections, that's a huge, huge task.
Yeah.
So is literally just perfusing the brain with glucose and all these anti-death agents and all that, is that really going to be enough to form those connections? And then once those connections form, is that the point where then consciousness starts to come back and all these higher global functions start to return to the brain?
Yeah, but it's like, what's the point in having activity in a neuron if it's not doing its function and communicating to the neuron? Like, what's the significance of the activity?
Maybe if you could isolate that function to a specific set of neurons in a specific set of the brain, you might be able to restore some sort of function. But
that would still require them communicating.
Yeah. But globally, if you're getting this connection back and these networks are forming, that would also now be treading on the ethical grounds of this, where they're saying we're not trying to bring back consciousness.
Yeah. Maybe it was because they were too cold.
Maybe.
Like, I don't know anything about kind of how memories are stored in the brain or anything like that. And I was just wondering, like...
Neither do scientists.
Right, okay.
That's still a debate.
So, because I'm just wondering, like, if there's any... Say these pigs, before they put the perfusion in, did you say it was like 4 hours after they died they put them? And whether there would be... If you could reanimate these neurons in their
Are those
structures...
memories still there?
Yeah, would the memories still be there? Because I guess the neurons are still in their forms that they've made, so they'd still go along, like, the same pathways, I guess. But... Is the form of that pathway itself like a kind of inscription of a memory, or not a memory, but like a
very, very good question. I don't know.
Where is it that memory? They think memories. Don't they have an idea? Where it's like they might be.
There's several parts of the brain, I think, right? Like there's the hippocampus is one of them.
Yeah.
But
when you lose memory, isn't it because those parts of the brain have been damaged?
Yeah.
So then, would it depend on the amount of damage that happened?
Yeah.
Before it was reperfused.
Yeah. But then I guess the question here though is, once you've lost all activity in the brain, is that all gone now?
Yeah. Because is that the extent
of
the
damage? It's a very good question. I don't know. Memory is a really deep, heavy field that is very specific. And actually, I can't remember what episode it was that I did a paper on memory that it was basically still saying it was basically a new concept of memory, and so till this day scientists are arguing about how memories are formed and how they're stored. So we still don't know the answer to that. We don't know the answer to the basic question behind that to even be able to begin to answer this. That's one of the problems of neuroscience. We know so little.
Is it like, so obviously the big like book that this makes me think of is obviously like Frankenstein by Mary Shelley, and it's been ages. I did it for like GCSE English, I think. It's been so long since I read it. But it's weird because I think in the book, and it's probably wrong, but I think in the book, kind of the bit that Frankenstein puts in the monster to like make him human or make him live, I think in the book it's a heart, and then in the film, a bit later, obviously, is in the film it's a brain that they put in. So already there was that kind of like shift from like thinking what constitutes a human is a heart to what makes someone human is a brain. But then I can't remember from the book, but I don't think the monster has any memories of of whatever the brain was before, you know. So it's this brand new living thing that comes out of nothing, I guess. Because if that brain in Frankenstein had retained it, you'd have expected the monster to wake up
and be like, oh, it's dinner time.
Yeah, exactly. Yeah.
Go.
So what do you think about, in terms of, you know how in this article that I read they were talking about this, we now might need to rethink what constitutes someone being dead or someone being brain dead. What do you think?
So I know that there's a lot of legal discussions going on all the time, isn't there, about what constitutes brain death, or death actually, just like normally, because and I guess it's often linked to the debates surrounding euthanasia and also life support and kind of when, ethically speaking, it is okay to turn someone's life support off, or when. I don't know. What are your thoughts on it?
I mean, I don't know. I'm quite black and white about these sorts of things, though. So I'm kind of like, once you're dead, if you've had to perfuse it to bring it back to life, you're still dead.
Yeah, I think it all comes down to quality of life. Like if you're being brought back to life and you're literally just like a brain lying there.
But in this, you're not even brought back to life. Yeah, some of your cellular functions have come back, but there are lots of cellular functions that don't die just because you've died. I mean, like, muscles still fire, and I don't know. I don't think that unless they manage to bring back some sort of network of neuronal activity or some sort of consciousness, I don't think there is a debate here about whether this brain is now considered not as dead as before.
But I guess if they did, they'd have to say that the brain is only dead if it could not be brought back to life by Bex.
Yeah. Yeah.
Lots to think about.
Yeah. It's really difficult.
It's not going to go through.
When I saw this paper, I was kind of like, this would actually be really great with you because it's got so many ethical
considerations.
Yeah.
I'm just like, I don't know. I'm just kind of. if you were somehow in that brain and you could see yourself on, and you know, it was like 6 hours had gone by and the Becks had been put in or whatever, and they were about to stop doing it, I don't know, I'd still be kind of like tapping on the glass of
your eyeballs.
Oh yeah, being like, please, no, give the Becks another chance.
Yeah.
Oh god, it's such a difficult question. Yeah, what?
I don't know.
This is going to sound like a very sad comment, actually, but like sometimes if I am feeling really alone, and this relates to nutrition, actually,
Go on.
I think sometimes if I'm feeling really, really alone, and I think this does relate to the question of what constitutes a dead body, I sometimes think, well, actually, I'm not alone because my body is more than one thing, and one of the things that I think when I'm thinking that is about the gut bacteria.
No way.
And I'm genuinely I just genuinely kind of think, okay, we're like working as a team to keep this body alive. It's not just me, and also it's not just, you know, what, like the me that is my brain or whatever. It's like, you know, there's loads of, it's the gut and back to the gut again, but I mean, it's like loads of different things. Your body's kind of like a very like plural object. I think.
God, you're so philosophy. I
really like that sentence.
But
your body is a very plural object.
But then it just like
use that
genuinely makes me feel less alone about the gut bacteria.
That's really interesting, because as well, when we started doing the podcast, I know I did a few gut bacteria papers. Like, I liked to have what do you call them? Analogies about the gut bacteria, and my favourite one was
soldiers.
The soldiers, like they're all the soldiers that are in your gut, and they're like protecting shit and stuff. And
that's interesting, because like when you're in school, and well, at least when I was, and like in primary school, I mean, and learning about the body, you learn the body through all of these kind of picture books that put it in loads of different metaphors, and those metaphors are either military, and it's always like obviously only men doing this. It's like obviously like pictures of like soldiers, or
no gender equality whatsoever,
or workmen, you know, in like overalls with like spanners and whatever, doing this thing. And so and then, like, you know, when you those TV shows where it like goes on a journey through your
like
your
the
body.
Magic School Bus.
Yeah, exactly.
Oh, I love that show. Yeah. That was my favourite.
And that's again that idea of like the body having loads of different things doing it,
going
you know,
on.
like, and you meet. It's kind of an adventure, and there's someone new in the heart room, and there's like a different type of creature in the lungs, and this kind of thing.
I mean, and then even like what's the movie where the emotions are little creatures?
Inside
Out.
Yeah.
Even in your brain and your emotions, they're all little beings.
About the bacteria again. Different type of bacteria this time, but what I find like a lovely image is this. You know, like you've got geologically. I was reading about this layer that they've found, or they always knew about it, but they didn't know much about it, or something like that. There is this layer of the earth that is just bacteria, and it's like quite deep down. Do you know about, like, because I haven't I don't know much about it at all.
No, I don't know much either, but I've
thought
heard
of it I find really like fascinating.
Because they've probably existed there for like an inconceivable amount of time.
Yeah, and it seems like they don't exist with the surface. There's
just this whole other community going on down there.
But I mean, I guess there's a layer of bacteria on everything, though, right? Like, there's just everything is covered in a layer of bacteria.
Yeah.
So they are sort of
Unless
their
you're
own.
sterile.
Yeah.
Unless you're a germ-free mouse.
Such a nerd. But I mean, so then they're all their own little like.
Yeah, but I think the point is that I think these are supposed to be like crazy deep down. So deep that you wouldn't have imagined anything could exist there, but there's all these bacteria existing there.
Yeah.
What is full of bacteria, I will say, is a beard. And what I will say is that walking through London with a beard is like, I don't know, it's like
putting out a net.
A fishnet, yeah. And I will say, and like, I think this is why I have such dirty nails. Because every day I start the day with pristine nails.
Yeah.
And then by the end of the day...
But London's disgusting.
Yeah, London, yeah.
Does it freak you out at all, ever?
No.
To think about the bacteria.
So do you get...
Oh, no. Like, I've really made... Since the probiotic conversation we had earlier, I've really made my peace with the... And as I say, you know, like, it's, you know, this makes you feel less alone.
Yeah.
Bacteria. And, No, it never freaks me out. Bacteria would never
So
freak
do
me
you
out.
find that you've been getting sick more since you grew your beard?
No, not at all, actually.
No, because if anything, it's keeping the bacteria
away. That's true.
Oh, right, yeah, I never thought about it like
Yeah.
that. Or just like, if it is giving you more bacteria. I always think, bacteria-wise,
Yeah.
stimulate that immune system.
Yeah,
Yeah.
exactly.
Like-
that's the whole theory about why people are getting so much like autoimmune diseases and stuff,
Yeah.
because we all got so clean.
When,
and
yeah, when,
kids
I, when I
aren't
was a kid, like, my
exposed
parents would always be like, no, pick that
Yeah.
off the floor, you can still eat that.
Yeah.
Or
Yeah,
That's...
they were doing you they were doing you and your immune system a good,
And I still,
a good
you
service.
know, like this whole like 10-second rule thing. I mean, if it's been there for hours,
Yeah.
maybe no.
You're like,
But
no.
like, yeah, I still always just think, okay, it's on
Yeah.
the floor. Like,
Yeah, I give a 5
Yeah.
second rule.
Should we wrap it up?
Yes, this was a really interesting discussion.
It was,
Yeah, thanks
and I
so
would
much for
I
having
would
me.
love to continue
I've
it.
made about
Yeah.
10 pages of notes.
And I'm just so proud and happy that you wanted me to draw out a diagram. I'm just really happy.
Oh yeah, we need to take pictures of all your notes.
Oh, yeah,
for
definitely.
Instagram.
Oh yeah.
So, how did you find the paper overall?
This paper,
Yes.
yeah, really, really interesting.
Good.
Great.
Did you
Yeah.
learn some stuff?
Yeah, of course I did. I learned the
You learned the mitochondria.
the mitochondria.
The powerhouse of
I remember this from. I mean, I remember it from
Everyone does.
biology.
Yeah.
But I think. Okay, can I just see whether I can? I haven't drawn a cell since biology. Can I
Okay.
just see whether I can do it?
Go for it.
Okay, so we're gonna do the blob. It's
Okay.
not a plant cell, so it doesn't have a cell wall.
Okay, very good.
Okay,
So
it's got.
while Ben's finishing drawing his cells, should
Okay.
we say goodbye?
Yes, okay.
Okay. Okay.
So thank you very much everyone for listening. I really hope you enjoyed this. This was like a very, very good discussion that we had. I think it was very, very...
I liked the meat. Sorry.
It's okay.
Are you okay?
Yeah.
Okay, I'm sorry.
Yeah.
I'm sorry.
I hope we
Maybe
did-
that'll be a bruise.
No, it won't. It's not that hard.
Okay.
Oh, I do have...
So it was a nice. Oh Jesus Christ! It was a nice coming together of science and philosophy. I enjoyed.
I hope no one had too much of an existential crisis. There was a few going on in this room. So whatever you're listening to us on, please rate us 5 stars, drop us an email, let us know what you think. If you have any suggestions for papers in the future, let us know. You can find us on Instagram and Twitter on @layout.com, @layoutpodcast.
And you can write to us on layoutpodcast@gmail.com.
24 episodes in and we still can't get this bit right.
No, but that's because we're rushing it now.
Can you get Patch on in the future?
Patch,
Patch.
Patch, we love
Okay.
you, Patch.
Patch is our new sponsorship venture.
Okay,
Okay.
bye.
Bye. Patch sponsor us.
Bye, 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 bye.
Bye.
Bye,
Bye,
thanks for having me.
bye, 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 bye,
Bye.
bye, Ben, bye,
Bye. Bye. Bye.
Bye.
Bye.
bye.
Bye.
Bye. Bye.
Bye.
Look at all
I'm
the
ready
hummus
for
that
that
we
shit.
left for Demel and she didn't even eat it. We left it all for you. Curry wouldn't let me have any more.
I'm not here.
I suggested we tell you to bring your own. So now you have to eat every last drop of that hummus.
You have to lick it clean at the end.
It has to be spotless.